Hello students, welcome to the course on Animal Diversity 1, Biology of Non-Chordates, Unit 2. Let's learn the general characters and classification of the phylum Porifera. The instructor for this topic is Dr. E. Harini, Head Department of Zoology, St. Joseph's College for Women Autonomous, Visakhapatnam. Contents, learning outcomes, general characters of the phylum Porifera, and classification of the phylum Porifera. Learning outcomes. By the end of this lesson, students will be able to identify the defining characteristics of phylum Porifera, including their cellular organization, body structure, and habitat. Explain how sponges exhibit a unique canal system, and describe the roles of pores, choanocytes, and spicules in their physiology. Classify sponges into their major classes, Calcarea, Hexactinellida, Demospongia, based on the skeletal composition and structural features. The phylum Porifera, which represent the sponges, are one of the earliest branches in animal evolution, displaying the simplest form of multicellular organization while maintaining complex biological functions essential to the marine ecosystems. The general characteristics of poriferans. Sponges are among the most ancient metazoans, representing a pivotal stage in the evolution of multicellular life. Their body plan demonstrates the transition from unicellular to multicellular organization without developing true tissues or organs. They are primarily marine species, permanently attached to substrates, with a few remarkable freshwater representatives inhabiting the lakes and rivers. They have an asymmetrical body plan, where it ranges from asymmetrical to radially symmetrical, lacking the bilateral symmetry seen in more complex organisms. They are characterized by numerous pores or ostia for water intake and larger openings called oscula for water expulsion, forming an aquiferous system. Coming to the cellular organization, the cells display some degree of independence without forming true tissues or organs, representing a cellular grade of organization. A gelatinous matrix between the outer panacoderm and the inner choanoderm, containing amoebocytes, spicules, and spongin fibers. The internal skeleton is composed of spicules, which are calcareous or siliceous, or spongin fibers, or both, providing essential structural support. The flagellated choanocytes create water currents and trap the microscopic food particles, serving as efficient filter feeders. And they are capable of both asexual reproduction through budding and gemmules and sexual reproduction with extraordinary regenerative abilities. The general characters. These are multicellular organisms with cellular level of body organization, no distinct tissues or organs. They are all aquatic, mostly marine, and a few are freshwater forms. They are solitary or colonial, and all are sessile. The body form is vase-like, cylindrical, tubular, cushion-shaped, many-branched, etc. The body exhibits radial symmetry or no symmetry. The body wall comprises of the outer pinacoderm, which is the dermal epithelium, the inner choanoderm, the gastral epithelium, and the gelatinous non-cellular mesenchyme in between. The mesenchyme consists of skeletal elements and free amoeboid cells. Cells loosely arranged do not form a definite layers, hence regarded not truly as diploblastic. Body with many pores or ostia, canals, and chambers that serve for the flow of water. One or more water exits or oscula are present. The choanocytes or flagellated collar cells usually line special chambers, and the sponges are the only metazoans having the choanocytes. The skeleton of calcareous or siliceous spicules of protein spongin fibers, or of both, or absent. Digestion is intracellular, no respiratory or excretory organs. Contractile vacuoles are present in some freshwater forms. The nervous system is primitive with neurons arranged in a definite network of bipolar or multipolar cells in some. All sponges are hermaphrodite, but cross-fertilization is the rule. Asexual reproduction by buds or gemmules. Sexual reproduction is by ova and sperms. All the sponges show remarkable regeneration power. The cleavage is holoblastic. The development is indirect through a free-swimming ciliated larva, the amphiblastula or parenchyma. The canal system represents one of the distinctive features of the sponges, and this system is an intricate network of channels and chambers which performs multiple vital functions simultaneously. Water enters through the numerous microscopic ostia, passes through the increasingly complex canal systems lined with choanocytes. They accumulate into the central spongocele and finally exit through the osculum at the top. This unidirectional flow ensures efficient filtration, and the entire system functions like a biological pump powered by the beating of the flagella of the choanocytes. And these water currents, whatever are entering through the ostia, which go into the canals, they carry oxygen for respiration, transport food particles for nutrition, they remove metabolic waste, and also facilitate reproduction. So this is the uniqueness of the canal system in sponges. The skeletal support. The internal skeleton of sponges provides structural support, protection from predators, and serves as the primary characteristic for classification. The two main types of skeletal elements exist often in combination. The spicules, which are microscopic, needle-like structures, they form the rigid component of the sponge skeleton. And these remarkable structures are secreted by specialized cells called sclerocytes, and they can be composed of either calcium carbonate or silica. Those which are having calcium carbonate are the calcareous sponges, which are characteristic of the class Calcarea, and siliceous spicules are characteristic of the class Hexactinellida. So the classification by structure, the spicules are of different kinds. The monoaxon spicules have a single axis with two ends. Triaxon spicules, they have three perpendicular axes, characteristic of the glass sponges. The tetraaxons have four rays from a central point. The polyaxons have several rays radiating from a single point, and they are star-like in appearance. And the flexible protein fiber providing structural support, characteristic of the demospongia. The spongian fibers form a network that gives sponges their characteristic soft, compressible texture while maintaining the structural integrity. In some commercial sponges, the mineral spicules have been completely replaced by spongin, creating familiar, soft, absorbent materials used by humans for centuries. Classification of Porifera. The sponges are primarily classified based on the composition of the spicules and skeletal fibers, and they are classified into three main classes: Class 1 Calcarea, Class 2 Hexactinellida, and Class 3 Demospongiae. The Class 1 Calcarea, the spicules are made entirely of calcium carbonate. These can be monoaxon, triaxon, or tetraaxon in structure, exclusively marine, and these are usually small and vase-like shaped. Example are Clathrina, Sycon, and Grantia. Class 2 Hexactinellida are usually made of siliceous spicules with characteristically 6 rays, and the skeleton often forms a fused lattice-like structure, exclusively marine forms, predominantly found in the deeper waters, and the famous example is Euplectilla, the Venus flower basket. The class 3 Demospongia contains siliceous spicules. They are never six-rayed or spongian fibers, and by far they are the largest and most diverse class, comprising nearly 81% of all the known sponge species, which includes both marine and freshwater species with enormous variation in form and habitat. Class 1 Calcarea, the calcareous sponges. These calcareous sponges represent the simplest class in terms of skeletal composition, with spicules composed entirely of calcium carbonate. They are exclusively marine, and characteristic spicule morphology makes them readily identifiable. The calcium carbonate spicules provide rigidity but are relatively soft compared to siliceous structures, and the spicules can dissolve in acidic conditions, limiting their fossilization potential. The spicules, coming to their morphology, display three main types: the monoaxon single axon, triaxon 3 axis, and tetraaxon 4 rays. And these shapes aid in classification and reflect the different mechanical properties. They are mostly small, vase-like tubular forms with a simple osculum or a single osculum. The body wall thickness and canal system complexity vary between simple asconoid and more complex syconoid and leuconoid structures. The examples are Clathrina, which displays a delicate lattice-like structure, Sycon, which is formerly Scypha, which shows an urn-shaped or a pot-shaped body with finger-like projections, and Grantia, which resembles Sycon but typically inhabits the temperate waters. Class 1 Calcarea comprises of order 1 Homocoela and order 2 Heterocoela. And here you can see the structure of Leucosolenia, a colony. You can also see the uppermost opening, which is the osculum. You can also see the images of different kinds of spicules, the monoaxon spicules, the triaxon, the tetraaxon. And here is a picture showing the body wall, where you can see the pinacocytes, the amoebocytes, the spongocele or the body cavity clearly. Class 2 Hexactinellida, the glass sponges. These sponges are commonly called as glass sponges. They are characterized by the spectacular siliceous spicules with 6 rays. And these spicules fuse to form an intricate glass-like lattice that provides both strength and flexibility. The fused skeleton usually forms a vase-shaped or a cup-shaped body. And these sponges grow slowly but can achieve a considerable size and longevity. Some individuals are estimated to be nearly over 10,000 years old, and these are exclusively marine forms, deep dwelling forms, which inhabit the deep waters often below 200 meters. Their fragile glass-like skeletons would be easily destroyed by the wave action in shallow waters. The two examples here you can see Hyloneema. This Hyloneema is commonly called as the glass rope sponge. And here you can see that this is the upper body which is having pores or ostia. And this portion is having the symbiotic polyps on it. And you can see this long white hair-like structures which are siliceous spicules, and these siliceous spicules are long spicules, and these are used to anchor the sponge in deep sea waters, and the upper body will elevate outside the water and the rest is held on to the sea bottoms. The other example you can see here is Euplectella, which is called as the Venus flower basket. And here in this Venus flower basket, this is usually one of the most beautiful glass sponges, and the dried specimens are usually displayed in museums. And these species of Euplectella, they exhibit a commensal association with certain species of shrimps. And whenever the young male and female shrimps enter the spongocele of the sponge, they stay there, and after growth, they are unable to escape through the minute sieves which are present on the body. So they lead their entire life within the sponge, feeding on the planktons, whatever are brought by the water currents. And the sponge with its imprisoned shrimps, it is like till death do us apart, and this is one of a very famous gift in Japan. The class Hexactinellida includes order 1 Hexasterophora and order 2 Amphidiscophora. Hexasterophora, the example is Euplectella, and Amphidiscophora, the example is Hyloneema. Class 3 Demospongia represent the most successful and diverse sponge class, encompassing approximately 81% of all the described sponge species. The remarkable diversity reflects their ability to colonize virtually every aquatic environment. They are found in all marine environments, and few of these demospongians even include freshwater species or freshwater sponges. They may contain siliceous spicules, or they are made of spongin fibers, and many species lack spicules consisting only of spongin network. This flexibility allows adaptations to various environmental conditions. They exhibit extraordinary variation in the morphology from encrusting forms that cover surfaces to massive barrel-shaped sponges, and the body structures range from a simple asconoid to a highly complex leuconoid canal systems. The example you can see are the Cliona or the boring sponge, and you can see here the boring sponge, which is made out of small pores. That is why it's called as the boring sponge, which erodes the calcareous substrates. And here is another example of spongilla, which is the freshwater species. Class Demospongia includes subclass 1 Tetractinellida, which includes 3 orders. Subclass 2 Monoaxonida, which includes 4 orders, and subclass 3 Keratosa. Here the example is Euspongia and Hippospongia. Let us look at some remarkable features of the poriferans. They are ecosystem engineers, since they play a crucial role in marine ecosystems as filter feeders, processing enormous volumes of water daily. They remove bacteria, organic particles, and dissolved organic matter, effectively functioning as natural water purifiers. They are habitat creators. Large sponge colonies create complex three-dimensional structures that provide shelter, feeding grounds, and nursery areas for numerous marine species from small fishes to crustaceans. They have evolutionary significance. Their simple structure belies complex biology and an extraordinary evolutionary history spanning over 600 million years. Understanding sponges provides insights into the origins of animal multicellularity. Despite their morphological simplicity, they exhibit sophisticated cellular coordination, remarkable regeneration abilities, complex life cycles. Their importance extends beyond ecology to biomedical research, as sponge-derived compounds show promise for the pharmaceutical applications, including antibiotics and anti-cancer agents. Understanding the general characters and classification of Porifera is fundamental to appreciating their ecological significance, their evolutionary journey, and potential contributions to human medicine. These ancient animals continue to surprise scientists with their complexity and adaptability. So let's summarize about the poriferans. The porifera are simple multicellular organisms, mostly marine and sessile, with a porous body and canal system that allows water to circulate for feeding, respiration, and excretion. They exhibit a cellular level of organization without root tissues or organs, and their body is supported by a skeleton of calcareous or siliceous spicules or spongin fibers. The nutrition is obtained by filter feeding through the choanocytes, with digestion occurring intracellularly. The sponges reproduce both asexually by budding, fragmentation, and gemmules, and also sexually, usually being hermaphroditic with internal fertilization and a free-swimming larval stage. They lack a nervous system and sense organs, and they depend on simple cellular coordination, but yet play a vital ecological role as filter feeders and habitat providers in aquatic ecosystems. These are the references for the topic discussed so far. Thank you for your patient listening.